In this lecture, we will explore the characteristics of eukaryotic cells. We've talked about the prokaryotic cells already, and the major distinguishing characteristic of the prokaryotic cells was the lack of a membrane-bound nucleus. So when we switch gears now and talk about the eukaryotic cells, that's going to be our hallmark, that true membrane-bound nucleus. Eukaryotic cells include everything except for bacteria and archaea, so that's the algae, protozoans, fungi, plants, and animals. So here we have this basic cell here, this basic eukaryotic cell, and right away you notice that it is much more complex than the sample bacterial cell, the prokaryotic cell we looked at in the previous video. You see a lot more structures inside the cell than we had with the prokaryotic cell. So this is just a generalized cell here, okay? Keep in mind that we use this as a model, that not all cells are going to have all of these parts. But what all eukaryotic cells do have in common is the nucleus. And you'll notice that there is a nuclear envelope. That nuclear envelope is very similar in structure to the overall structure of the cell membrane, a phospholipid bilayer. There are pores within that nuclear envelope that allow RNA to be able to leave the nucleus. We have in the center of the nucleus the nucleolus, there, this kind of darker staining structure. And then we have all of these other membrane-bound organelles. So we have the endoplasmic reticulum, both rough and smooth. We have the mitochondrion here. We have the Golgi apparatus here. We've got lysosomes. We've got peroxisomes. We have centrosomes here. We still have ribosomes, just like we saw in the prokaryotic cell. We also have cytoplasm that's going to fill in the areas. We can still have flagella, like we saw on the prokaryotic cell, but we see something a little different here on the external surface. We have cilia. We don't find the pili or the fimbriae on the eukaryotic cells like we did on the prokaryotic cells. The other thing that's going to be different when we look at eukaryotic cells is that on average our size of eukaryotic cells are going to be larger, and the eukaryotes themselves are going to be larger. Okay. Now, if we kind of take a look here, just like we saw with bacterial cells with the prokaryotes, we're going to have different cell morphologies that we'll see with the eukaryotic cells. We'll have some that are more circular in shape, some that are oval in shape, some that live inside our cells, some that kind of have this stringy appearance or this bell or cup-like appearance. So we can still have a variety of different shapes with our eukaryotic cells, just like we saw with the prokaryotic cells. So again, the main structure that sets the eukaryotes apart from their prokaryotic counterparts is the nucleus and the appearance of these other membrane-bound organelles. This is a fluorescent microscope image where you can really see a really good clear picture of the nucleus with this nuclear lamina around it. So you can see that there is a well-defined border between the contents of the nucleus and the contents in the rest of the cell. I pointed out the nucleolus when we were looking at the overall picture of the cell, that it is this dark staining region within the nucleus, and this is where rRNA or ribosomal RNA assembly occurs, as well as the formation of ribosomes. Now, we saw ribosomes in prokaryotes as well, and the ribosomes are the sites of protein synthesis, and they have the same function in both prokaryotic and eukaryotic cells, but the structure is different. In eukaryotic cells, they are 80S ribosomes, whereas in prokaryotic cells, they are referred to as 70S ribosomes. So even though they have the same function, they do have different structures. Also, something that is different within eukaryotic cells is that associated with the endoplasmic reticulum here, we will have ribosomes, and these are called membrane-bound ribosomes. They are attached to the endoplasmic reticulum, and we call that the rough endoplasmic reticulum. And what happens is that when these ribosomes produce their proteins or synthesize their proteins, they then drop those proteins and they can be transported throughout this membrane system here. Some of the organelles, other organelles that we find in eukaryotic cells is the Golgi apparatus. I refer to the Golgi apparatus as the FedEx or the UPS because the Golgi apparatus is going to receive substances that are being produced by the cell. It's going to package those substances in little vesicles and then transport those where they need to go, either somewhere else in the cell or most likely outside of the cell to another cell in the body. We have lysosomes, which are basically little sacs of digestive enzymes, and these enzymes can be used to break down food particles or damaged organelles or ingested, bacteria or other cellular debris, or they can be released and kill the cell if it becomes damaged. Peroxisomes are similar to lysosomes in that they are little bags of enzymes, but peroxisomes produce hydrogen peroxide, and that's how they were originally named. But peroxide helps to break down uric acid, which is a waste product of the cells, amino acids. They have the enzyme catalase, which can degrade hydrogen peroxide. And this is something that we will see later on in the semester, in that some organisms have catalase and others don't, and that's something that we can use to help us distinguish between different types of bacteria. Within eukaryotic cells, we also see this cytoskeleton, which basically is what provides the framework and the structure to the cells. We have microfilaments, intermediate filaments, and microtubules that all work together to produce this cytoskeletal network. Now, remember that our cell membrane is a phospholipid bilayer, and what we're going to see is that some of these filaments that make up our cytoskeleton can be attached to the cell membrane that allow some flexibility in the cell membrane and the cell itself. Another organelle that we see in eukaryotic cells is the mitochondria. The mitochondria is the powerhouse of the cell. This is where cellular respiration occurs, specifically aerobic cellular respiration. And so this is where we take sugar and use oxygen to produce ATP. We'll talk more about the process of cellular respiration later on in the semester. We also find chloroplasts, and these are found in plant cells and algae cells. They are the sites of photosynthesis. So we only find chloroplasts in those cells that perform, the eukaryotic cells that perform photosynthesis. Now, again, taking another look here at our plasma membrane, we do see that phospholipid bilayer with the associated proteins and glycoproteins, carbohydrates, glycolipids. So the overall structure is going to be very, very similar between prokaryotic cell membranes and eukaryotic cell membranes. The membrane transport processes are also very similar. We have simple diffusion, we have facilitated diffusion, and active transport that occur in both prokaryotes and eukaryotes. However, eukaryotic cells can also perform endocytosis and exocytosis. Endocytosis is a way to take matter into the cell, and then exocytosis is a way to have matter leave the cell. There are couple different types of endocytosis. We can have phagocytosis, which means cell eating. So this would be the transport of solid particles. And then pinocytosis, or cell drinking, would be bringing in liquid particles. Now, some eukaryotic cells have cell walls. Plants have cell walls. Fungi have cell walls. Algae have cell walls. And some don't. Protozoans and animals don't have cell walls. And the structure of the cell wall varies between each of those groups. Now, if we look at the external structures, we have flagella and cilia. The structure of flagella is different between prokaryotes and eukaryotes. However, the function is the same. The primary role of flagella, no matter what type of cell we're talking about, is locomotion, movement. Now, cilia can have different functions depending on what type of cell they're in or they're on. And cilia can be used for movement. They can beat all at once and be used for movement, or they can be used to move food particles or other debris past the surface of the cell. So it really depends on what type of organism and what type of cell we have that we're looking at to determine the function of the cilia.